since we've all gathered with AAG and with spring break, so it's good to see you all here again. We are going to start out with some department announcements, and then we'll move into our diversity share before we move into our speaker today. So first off, we want to see if anyone has any announcements of any shout outs they want to give, any upcoming events you want people to know about. I'll open the floor to everyone else.
I just want to announce that James Bryan accepted an offer for a PhD program at UNC Chapel Hill yesterday. Congratulations, James. Really proud of you.
Yay, that's great. Congratulations, James. And I'll give another shout out to Nazneen Sultana, who got the Qualitative Methods Specialty Group Award at AAG for her research on the political ecology of climate change. If you didn't see the email go through, so big congrats to Nazneen and to everyone, honestly, who presented at AAG, represented UD really well there. Any other announcements? We do have another seminar next week on Wednesday. Salim, did you want to say anything about that?
Yes, this is a special seminar we are having because we couldn't fit into the regular schedule, but there's a faculty member from University of Notre Dame who is spending a year at the Princeton Institute for Advanced Study, and she is a specialist in water anthropology. She's got a PhD in anthropology from University of Chicago and works on the anthropology of water and bureaucracies of water. And so she's going to talk about the recent flooding in Pakistan and also discuss a book she's working on. I think it'll be really good for the graduate students also because she's converting part of her dissertation into a book. And she'll talk about how to do that. And she'll discuss, I think, one of the chapters of her book also. So it's Wednesday, I think at 2 o'clock.
Yeah, and 2:18.
Yeah.
And online as well. Great. Any diversity shares? This is an opportunity for us to share resources, events, other opportunities that increase our awareness of DEI efforts on campus and in the community. Any diversity?
Well, I guess I can say in terms of diversity, I was in Greenland recently for field work for an NSF project. And what I found out there in terms of diversity experiences was that the term Eskimo we use sometimes, Eskimo is considered offensive by many of the people there, and they prefer Inuit as the term, or in their case, Greenlandic. But I grew up hearing the term Eskimo. I don't know if others did too, but it was something I found out was considered offensive. They prefer Inuit.
Great, thanks, Salim. I'll also put a link in the chat if folks haven't seen the reports from the AAG. They are presenting data by race and ethnicity and gender identity in the previous months. If folks haven't seen some of their reports on the state of the field, if you're interested in seeing kind of where geography has been and where we're going in terms of DEI efforts, those reports are really helpful as well. Okay, anything else? Okay, with that, I will pass it over to Kevin, who's been part of the seminar organizing committee this semester, and he's going to introduce our speaker today.
Thank you. So our speaker today is Professor Allison Steiner. She's the Associate Chair for Graduate Studies at the University of Michigan and has co-appointment at the Department of Climate and Space Sciences and Engineering and the Department of Earth and Environmental Sciences. She's also a member of several international and national scientific boards, the Board on the Atmospheric Sciences and Climate and the National Academic of Sciences, Board on Atmospheric Sciences and Climate, the Board on Atmospheric Biosciences, American Meteorological Society and a member of the Scientific Steering Committee at the Global Emissions Inventory Activity. And she's also the president-elect at Atmospheric Sciences section of the American Geophysical Union. She leads the Steiner Research Group, which studies the biosphere's interactions with the atmosphere and how it impacts atmospheric chemistry with regards to climate. Please join me in welcoming her today as our speaker.
Thanks for having me. I'll go ahead and share my screen now, and 1 second. And can everyone see everything okay? All right. Yeah, so thanks for having me. As you just mentioned, I'm in the Climate and Space Sciences Department at the University of Michigan, and I'm an atmospheric scientist who's done a lot of work on regional climate and atmospheric chemistry. And so the talk I prepared today was one that I know your department kind of covers a broad aspects of geography, both the physical system and the social system. So I tried to pick a topic that at least would provide some interest for everybody, even if you may not be that interested in the atmospheric sciences or the climate part of it. And that's talking about the atmospheric life cycle of pollen. And so I got interested in pollen a couple of years ago because of sort of how we talk about aerosols in the atmosphere. And so again, as an atmospheric chemist, we often categorize aerosols as being, particles, gas or liquid in the atmosphere, liquid or solid particles that are suspended in the gas of the atmosphere. That's the definition of an aerosol. And they can have different origins. So they can be primary, which means they're a direct source coming directly from, in this case, the terrestrial biosphere, or they can be, in many cases, secondarily formed by chemistry in the atmosphere. So the primary versus secondary is referring to the origin of emission. And then the biological is referring to particles that are coming from terrestrial sources that are biological in nature. And so here are 3 examples of different types of primary biological aerosol particles. Sometimes people use the acronym PBAP. And they really range in size pretty broadly. And they're showing you some SEM images of these tiny particles that ended up getting lofted into the atmosphere. So bacteria on the very small scale of around a micron, fungal spores are a little bit larger, about 4 to 10 microns, and pollen, which can be on the order of 10 to 100 microns. And typically atmospheric scientists haven't been that interested in pollen. And predominantly the reason for that is because of the size. So this is showing a standard aerosol size distribution. One of the challenging things about aerosols is they range over several orders of magnitude in terms of size. And so when we look at different types in the atmosphere, there's so many different sizes, we often use a continuous distribution to represent them. And from the climate perspective, the ones that are sort of the most important are ones that we kind of see in this accumulation in the ultrafine mode. And part of that is because they have a climatic impact. They can scatter incoming radiation, and they can also influence clouds. And then as well, the smaller ones have a health impact. And we often think when we regulate particles, we're regulating particulate matter, which is usually less than PM 2.5, which is sort of shown in this range right here. And so the PM 2.5 is important because those particles are the ones that can make it past your respiratory defenses and get deeper into your lungs. So as a result, we don't really often pay many attention to the coarse particles, which are over here. And so pollen is the kind of particle that sort of resides in this coarse mode. And so there hasn't been a ton of attention paid to it in terms of its relevance in the atmosphere. But I got interested in studying pollen because once we put it in the atmosphere lots, it can actually be processed within the atmosphere and we can change its size. So it can undergo different rupture processes in the atmosphere. And I'll come back and talk about these mechanisms a little bit later. But these are some SEM images that show you, here's an ambrosia or ragweed pollen grain. And these tiny little particles that are coming off the other side are essentially these sort of inner parts of the pollen grain. It's mainly based up of carbohydrates. And so these tinier particles are one that are now suddenly climate relevant and also a lot more health relevant as well. Fungal spores can do the same thing as they can rupture. And so essentially by being in the atmosphere, they can be processed and we can change the size of these particles that influence their importance. So what I was going to talk about today is how we are trying to, essentially include PBAP within Earth system modeling frameworks of how we can model these emissions of bioaerosols. And specifically, I'm going to focus on pollen and what I sort of call the atmospheric life cycle in terms of how we emit different pollen, how they might be ruptured, how they could influence clouds, and then also be returned back to the surface through processes of dry and wet deposition. So again, like why do we care about pollen? This is a figure from North Carolina in 2019. During one of the years, people were sort of talking about the pollen apocalypse. So there are some years where we have very, very high pollen concentrations in certain regions that can cause these sort of large yellow clouds that form that we can actually see optically with our own naked eye. And I'm sure everyone's also familiar with where you're living in Delaware that, you know, you come out of your house and your car is covered in pollen. And so, you know, part of it is that we can see it. So therefore, we should be able to think about it's got to have some importance. And secondly, it's a big driver of seasonal allergies, which affect over 30% of the US population and more globally. And so we should really be able to develop better predictive models for pollen and understand some of these meteorological and climate drivers that influence emissions. So this is sort of how I started getting interested in pollen. I had done emissions of trace gases from the terrestrial biosphere before, and then started working on these primary biological particles. And it is kind of captivating to look at some of the different SEM images of pollen. So here's a picture of a grass pollen grain. A lot of people are allergic to grass. Grass is on the order of around 20 microns, and pollen are usually identified by these sort of what people call the exine, or the harder outer shell, which has different physical features that allow it to be identified when people are looking at it under microscopes. Here's an example of ambrosia pollen. It also has a very distinctive exine, this sort of spiky look that allows it to be easily identified. And again, a lot of people are allergic to ambrosia. As I'll show, it has a different seasonal cycle, so you might experience that in the fall. This is typically, ambrosia is typically associated with what we call hay fever. And then finally, birch is one that's coming from deciduous trees. Again, you can see the shapes of these grains are slightly different. And birch is something that a lot of people are allergic to as well. And finally, one of my favorites is pine pollen. So pine pollen is really, really large. People typically don't think there are many allergies to pine pollen, but it can cause these types of events like what we see in North Carolina off to the left. And pine is really interesting because you can get these very large air bladders that are forming called sacci, which actually influence the buoyancy of the particles, which allows it to be lofted more highly into the atmosphere. And so it's kind of an interesting structure that the pollen has developed to increase its transmission. So just as a reminder of why pollen is out there to begin with, this is a way of reproduction in plants. And so this is showing you from a classic biology textbook. We have a female component of the plant, and here we have our male component of the plant and the anther. And the pollen is produced originally in the anther. It undergoes meiosis and mitosis to create the gametophyte, which is actually our pollen grain. And within the pollen grain, there's generally two cells. So this sort of diagram is showing you the exine right here, and that's how we identify pollen. But on the interior of pollen, it's mostly just cytoplasm with two different cells in it. One is the generative cell, which is carrying the genetic material. And then the second is the tube cell. And what the tube cell does is really amazing. So when the pollen grain is near the female component, there's usually some chemical sensing that's involved in this, and it lands on the stigma here. The pollen grain will actually grow a pollen tube, which will grow from the top of the stigma all the way down to the ovary. And through that pollen tube, it'll deliver the genetic material and fertilize the egg. And so then that allows fertilization to occur. So typically, when we look at these exines, when I talked about the rupture earlier, the exine is rupturing in the interior components of the cytoplasm, and it's predominantly carbohydrates and starch. That's what's really coming out of this pollen grain when we see the rupturing. And this is happening more as an abortive process. It's usually typically before the pollen grain reaches its intended target. So pollen can be transported in lots of different ways. We often think of the biotic mechanisms first, where we have bees as pollinators. That's called entomophilous types of pollen transport. You can also have zoophilous with animals. This is a picture of a Darwin finch here, showing you how birds are often also used as a way of trying to transmit pollen. But what I'm going to really focus on today, and again, this is because I'm coming from this atmospheric background, is what's known as the anomophilous pollen, or the wind-driven pollen. And this is a figure of this right here, showing you a pine tree. So the pine cone structures here are all carrying pollen grains, and they're designed to be dispersed. The pollen grains are designed to be dispersed by wind. So most gymnosperms, or coniferous trees, use anomophilous solely as their mechanism for pollen transport. When we look at deciduous trees, then they're often using a blend of either either anemophilous or in some cases the entomophilous types of reproduction. So when we look at this across the globe, sort of the biogeography as we call it, a lot of this anemophilous species, this is showing fraction of anemophilous species for all the different types of vegetation, they tend to be more concentrated in the northern mid-latitudes and up to the northern latitudes. In the tropics, a lot of the trees are using the biotic mechanisms because we have this really large abundance of bird and insect species in those regions. And so a lot of what I'll focus on is talking about over the continental United States, but it's important to note that these anemophilous mechanisms and these feedbacks with the atmosphere are really largely driven, by this wind-driven type of mechanism, which is in the northern, more common in the northern hemisphere. And also you can see it in the southern hemisphere as well, but just generally not around the tropics quite as much. So I'll sort of talk about pollen from two different perspectives. One is how we've started to develop a model, a mechanistic model for pollen emissions and how they'll change in the future. And then the second part, I'll focus a little bit more on the atmospheric side of things, understanding how pollen can change in the atmosphere and affect clouds and human health. So when we started working on this, there's really not, amazingly, given how many people have seasonal allergies, there's really not a ton of actual pollen counting stations across the United States. So you might have a pollen app if you have allergies, you might have an app on your phone and it'll give you like a geolocated pollen count, but the data behind that is very, very sparse. And so there's maybe on the order of 100 to 120 at any given time pollen counting stations across the United States, and they're run by the American Academy of Allergy, Asthma, and Immunology, or the A4I. And so they've been collecting this data, and usually they tend to be at allergy centers or at hospitals. They tend to be in urban areas where people are located because the counts are occurring because they're trying to help people manage their allergy symptoms. And so they count 43 different types of pollen at the genus level or the family level, depending upon what type of vegetation it is. And the plots here are showing you about a decade's worth of data from the A4I stations organized by region. And you can see a couple different things. So in the Northeast, which is no, Delaware actually fell in our categorization. Delaware is following in the Southeast. I'm not sure how everyone feels about that. But here you are located here. And so in the Southeast, we really see this very strong deciduous peak. So we're moving into that peak. You're probably getting close to hitting peak deciduous pollen. Interestingly, you can see the evergreen pollen is sort of happening a little bit earlier. And a lot of that has to do with some of the cypress species across the Southeast. And then you have grasses sort of occurring throughout the summer at much lower levels. And then here in the late summer, early fall, you'll see this ragweed peak, or that's the ambrosia I was talking about. So this is what we classically call hay fever, but obviously you can be allergic to pollen at any different parts and any time of the year. And the Northeast looks fairly similar. They have a much stronger deciduous peak. As we move into the Mountain West, you can see our stations are very sparse, and the coniferous trees dominate. And we see contributions of both coniferous and deciduous in the Pacific Northwest. So using this data, we wanted to try to develop a model that was spatially consistent for pollen emissions. And so this was work by two different graduate students over the past several years. One is Matthew Wozniak, who graduated a few years ago, and it was picked up by Ingzhao Zhang, who's a current student with me here at Michigan. And so we developed an emissions model that we could do, to be able to develop daily pollen emissions across the whole continental United States. And we base this on three different factors. So we're calculating the pollen emissions E. The first factor is what we call the production factor, which is essentially saying, so a plant sort of will determine, it'll allocate its resources to produce pollen at the beginning of the year. It'll put that pollen out and then it's done. So we sort of use some literature values of pollen grains based on the different types of vegetation that we saw from the A4I data from the counting stations. And so we developed production factors for each of these based on these literature values. So at this point in time, we're holding these as static. So here I'm showing you we have several different genera for the deciduous trees. We also group them together into one category, the deciduous broadleaf forest. And one of the reasons for that is it's known as plant functional types, which we often is used as a land cover classification in climate models. And so I wanted to make sure it was flexible to use when we had a lot of tree data, when we could actually go at the sort of genus level, or if we were down to the sort of more generic like deciduous trees level. We also use the A4I data with climate data to try to understand whether or not these production factors might be temperature dependent over time. And one of the things you can see for all the different types is there's a lot of variability. So for some types of vegetation, it seems like we might have a positive influence in production with increasing temperature, but there are also some that have a negative impact. And so one example I'll point out which is one of the pollen grains I showed earlier that commonly causes allergies, is birch. Birch seems to be a very climate sensitive species where it looks like pollen emissions may go down under warmer temperatures. This is still very uncertain because the data is very sparse and we don't have a lot of it and it's rather noisy, but these are some early estimates that we started including in the model. The second thing that we use is the area. So we're using flexible land cover data input where we can do it by plant functional type, which I'm showing here. So we could do it for 4 categories like deciduous trees, evergreen trees, grasses, and ragweed. And then we also have a version where if we have better tree data at the genus level, then we can also model across the United States at a better resolution. And then finally, we take these productions and we spread them out over the year using a phenological Gaussian curve. And so what that means is we're taking this sort of, a one-time production value, and then we distribute it over different days of the year based on the observed start day and end day relationship of temperature. So what does that look like? So when we use the A4I data, this is data from all the counting stations, for example, for birch that we have across the United States, we can look at the relationship between the prior year annual average temperature and the start day of year for the birch pollen season and the end day of year for the birch pollen season. And you can see that the correlations are fairly good. Again, just remember this is rather very sparse and noisy data, but we use that data to develop relationships between temperature and when we think a pollen season would start and end. And then we fit a Gaussian curve to that to describe this phenological curve for all these different types of vegetation. And so that goes into building the model. So let me give you an animation of what this looks like. So this is just showing you a sample year for 2001, and we have the 13 pollen types grouped by plant functional type. And we're doing this at a 25-kilometer resolution across the United States. Okay, so here we come into June. You can see here the ragweed as we move into late summer, it sort of starts to blossom across the United States. As we move into the winter, we can see the pollen start to decrease. And then the first thing we see coming into the out of the winter is here's that evergreen that's coming from cypress species within the Southeast. As we build into spring, here's our deciduous. And then here you can see our grasses coming out in the summer and moving up as temperatures get warmer. And so this sort of gives us a first start in terms of trying to understand how we can actually develop a spatially consistent pollen emissions model that is climate flexible, meaning that as temperature changes, and we also have included some precipitation changes as well, we'll see changes in those emissions. The next thing we did, and I'm not going to spend a lot of time talking about this, but I have the reference on there, and we got a lot of interest in this article that came out last year, was trying to understand how future climate would influence these pollen projections. And so with this, we used 15 different CMIP6 models. That's from the Climate Model Intercomparison Project that's run by the IPCC. And we compared a historical time period from 1995 to 2014 with an end of century simulation. And we did it for 2 different SSPs, or sort of future warming scenarios. And what I'll show in the next slide is from our sort of higher warming from the 585 scenario. So it's like sort of by the end of the century, we've been warming about 4.5 degrees globally. Hopefully we're not following that trajectory, but this was what will happen to the pollen if we do. And this is showing work from Ing Zhao's paper, which just came out last spring. And so this is showing you for those same regions I showed again. On the left-hand side, this is the historical amount of pollen we had, and depending, showing you for these different types of pollen that we model and what happens in the future, how it changes. And so overall, we see that the total pollen emissions are increasing by about 15 to 40%. And the other interesting thing we tended to see a lot more shifts in the distribution. So for example, one of the things that you can see in the Northeast is this sort of brighter blue color is quercus or oak, and we can see how much we expect oak emissions to change in the future as well. And so if any of you are interested in that, I can talk about that paper a little bit later, but we got a lot of coverage from that in the media last year as people were concerned about what this would mean for seasonal allergies. So that's where we are with the emissions model right now. There's still a couple things that we're hoping to improve. One, as I mentioned, the annual production factors. So, right now we're using literature values that we treat as I mentioned, we treat them as static. So we don't have any year-over-year variability in terms of that, unless we do these longer runs where we allow temperature to influence the pollen production factors. And at this point, we don't really have a great handle on what the biological drivers are of the interannual variability of pollen. We still don't really understand why some years the pollen might be higher than others. And that's something that we're hoping to continue to work on and disentangle. The second is a term we often use in the climate community about stationarity. And so you can see I showed you a linear regression between the start day and end day and the temperature. And so, one of the things that we often assume is when we forecast into the future and we use empirical relationships like that, we assume that the statistics that are happening in the historical period are also going to apply in the future. And so we don't really know whether or not those past statistics will hold. And so that's why we're hoping we might be able to develop better mechanisms for explaining, biologically why pollen would be changing from year to year. So the second part of the talk, I want to talk about how we've been putting pollen into atmospheric models and how we think it can be influencing climate. So this gets a little bit more into the atmospheric science part of the talk. And looking at this comes back to the same cycle figure that I started with, trying to understand how we can use three-dimensional Eulerian models to model this sort of what I call like the atmospheric life cycle. How do we emit it? How is it processed and ruptured? And how do we create these tinier particles, what we're calling the subpollen particles? How do they get transported and how are they removed from the atmosphere? And then finally, we're also really interested in how it's influencing clouds and precipitation processes, because these tiny particles can act as cloud condensation nuclei, meaning they can act as seed particles for clouds, as well as they can act as ice nucleating particles, which means that they can act as seed particles for ice clouds. So both of those processes we think are important. So one thing I'd like to come back to is this idea again of the atmospheric processing. I sort of started with this in the beginning, talking about the size distribution. And so here's the same SEM image that I showed of the pollen rupturing. And so why does this happen exactly? One of the thoughts, there's kind of two different theories about how and why pollen could rupture. I mean, the first is that we have, this is a figure from a recent paper that just came out in JGR Atmospheres. Here's our pollen grain. Okay, so the pollen wall is the exine that I talked about. That's usually what gives pollen its distinguishing features. And then we might have some cytoplasm in the inner part of the wall. Okay, and so as we put this pollen grain into moist environments, it's going to start to swell. Okay, and so as it starts to swell, we can see that movement of the exine. And eventually, as we have more and more water being taken in through the pollen into the pollen grain, because they do have these openings, okay, what are called apertures in the exine, which allow that pollen tube to grow. So water can enter. And ideally, the pollen grain was designed, sort of to be able to hydrate and dehydrate as it goes through different environmental conditions. But there can be conditions where it gets too moist for it and the turgor pressure inside increases, and that leads to the burst and this sort of rupturing and the release of these tinier subpollen particles. So this is often known as like the rupture process driven by humidity. So we can think that as we go into really humid environments or as we go into cloud environments, this type of osmotic pressure could cause the pollen grain to rupture. The second theory that's out there is about lightning. So lightning is also thought to deliver this sort of same kind of osmotic shock. And so other people have postulated that lightning itself could be responsible for the rupture of pollen grains. So then we come into this part about the clouds, right? So there's a couple ways that pollen could influence clouds. One is that the primary pollen itself, those large pollen grains, could actually act as what we call giant CCN. So generally CCN, we like them to be small, but there can be cases where large particles can act as a good cloud condensation nuclei. And that's really affecting what we call the liquid phase or the warm phase of clouds, where you're below the freezing level within a cloud. We also have speculated that ruptured pollen, those tiny subpollen particles, could also act as cloud droplets by CCN within the liquid phase of the cloud. And then as we get into the colder parts of the cloud, this sort of middle range is what people often call the mixed phase, because you'll have liquid and ice phase hydrometeors together, as well as into the purely ice phase, which is about below 38 degrees Celsius, that both the primary pollen grains and these ruptured particles could act as good ice nucleating particles. And that's what's sort of shown right here. And there's lots of different ways that ice can freeze. There's a couple different mechanisms. You can have what's known as condensation freezing. So if this is my tiny particle and it ends up in a cloud drop, then that can freeze. You can also have something called contact freezing, with if that little tiny pollen particle comes in contact with an ice crystal, it'll come in contact with a liquid drop, and that sort of touching of the two of them will change the dynamics and make an ice droplet. And then you can also have something known as immersion freezing, which is where you have the droplet here and it then forms an ice crystal in that way. Once you get up into the ice phase of clouds, then you can actually get what we call homogeneous freezing, which means you don't need any particle to freeze, but we can also see these other types of freezing processes important in the ice phase as well. And so this is kind of one of the questions that we wanted to try to explore, which is that when you put pollen particles in the cloud, how does it influence how these processes happen? So we started with the warm clouds, and several years ago, I worked with a colleague of mine, Sarah Brooks, at Texas A&M University in the Atmospheric Sciences Department there. She does work on cloud condensation nuclei and had the laboratory equipment so we could also start to test this out. And so we did some live pollen collection in the field down in Texas, and we made what we call like aqueous samples. So we take the pollen and you swish it around in water, and then you filter it out, and you end up with these solutions. And you can actually see like the colors of these solutions here. You atomize that to make little liquid droplets, you dry it, and then you size it, meaning like you select the size of the particles, and then you put it in an instrument called a cloud condensation nuclei counter, and you watch to see how the particles grow. And if they grow into cloud droplets over a range of different saturations, then that means that they're good cloud condensation nuclei. So we tested this with several different types of pollen. And so this is the size of the pollen on the bottom, and what's known as the critical supersaturation, meaning how much above saturation do you have to go in terms of like supersaturation of water to be able to allow this to occur. So the lower supersaturation you have, the easier it will be for a particle to act as cloud condensation nuclei. So as a reference point, we think of ammonium sulfate, this blue line here, as a very good CCN. It likes to form liquid droplets very easily, and it also takes up a lot of water. It's very hygroscopic, and you'll see those particles grow. In contrast, a dust particle doesn't really like to take up water, and we consider that to be fairly insoluble. And so its curve would look like this. So you'd have to get critical supersaturations of above about 1%, which means you're above 101% of supersaturation, which is relatively harder to get to in the atmosphere. These numbers are a lot easier. And so what we can see is that the pollen, we tested 6 different types of pollen, and all of them were actually, they're not as good as an ammonium sulfate, but they're much, much better than an insoluble particle. And this really suggests that pollen can, the subpollen particles that we generate by rupturing them in these vials can be a good cloud condensation nuclei. So then we wanted to test this out in models to see if this had an impact on precipitation, right? As we add aerosols to the atmosphere, it can have direct effects, but it can also have indirect effects on clouds. And so in this case, we used a regional climate model, and we included the pollen emissions I talked about in the first part of the talk, and we allowed it to go into the atmosphere. We allowed it to rupture at the surface. And then if it was in the atmosphere and the relative humidity was above 80%, then we assumed that 70% of those would rupture, and then they could act as cloud condensation nuclei and affect the formation of clouds. And so the idea behind this was, how much pollen do you need in order to be able to influence clouds and precipitation? So one big question about this, you can see there's a couple different parameters that we define. We define 80% relative humidity. We defined a fraction that's going to rupture. One of the bigger questions is, how many of these little tiny particles here are you making per pollen grain? And this is something we can do a sensitivity test in the model. I should say in our schematic here, we show it sort of like Pac-Man-like exploding, making lots of particles. And in fact, it's probably more like the other figure you'll see this rupture. So it's not really like a Pac-Man release of all these particles, but that was the best schematic we had at the time. So we can test this within the model. So we did this looking at the impact of pollen, of these ruptured pollen particles on warm clouds. So in the left-hand figure, I'm showing you the precipitation. This is springtime precipitation, and it's a multi-member ensemble, which means we run the model many times to try to take out the noise. And you can see sort of springtime precipitation concentrated along the East Coast. And then we look at the change in precipitation for 2 different pollen sensitivity tests. The first one, where we allow about 1,000 little tiny particles per pollen grain. So when it ruptures, it would release 1,000 particles. And then the other sort of higher sensitivity case is based on, if we rupture it, we release 1,000,000 particles. So the reason for the 1,000 and the 1,000,000 numbers, this factor of 3 difference, is when people did the SEM studies, the ones I showed in the very beginning, they would look at one pollen grain under the scanning electron microscope, the SEM, and they would change the osmotic conditions to make it rupture, and then they would count all the tiny SPPs. And when they would count them, they counted about 1,000. Now, if we were to do a pure mass balance and say, okay, this is the mass of the pollen grain, and if we were to make tiny particles, how many would it make? Then we get an answer of more of 1,000,000. And so the difference between the impact of precipitation of if we have 1,000 tiny particles per grain or 1,000,000 has a big impact on precipitation. So when we have about 1,000, it has pretty much no influence on precipitation. It's pretty much in the noise. When we have about 1,000,000 particles per grain, then we can see large changes in our springtime precipitation. And in the eastern part of the US, it was around 30%. And in some parts of the Intermountain West, it was up to 50% reduction in precipitation. So it was also just the total magnitude of precipitation as well as the intensity. So this is showing you precipitation binned by like the intensity in terms of millimeters per day. And so what we saw was fewer intense events of precipitation when we added lots of pollen, and more lighter amounts of precipitation, so essentially more drizzle. And this is pretty typical in terms of like when you add more CCN to models, how it can influence clouds. When you have a fixed amount of water and more CCN, the cloud droplets don't grow as quickly, and it tends to suppress precipitation. So one of the takeaways from this study is that we can really say we really needed to have a large amount of these subpollen particles in order to influence warm clouds. So then after this work, we were kind of interested in exploring this more. We were interested in exploring this more within a model we could run at higher resolution and look at more detailed cloud processes than we could in the regional climate model. And we also wanted to be able to test these different rupture parameterizations. So using a WRF chem modeling framework, so that's it. WRF is the National Weather Service Weather Research and Forecast model, which we often use for forecasting. And the chemistry version includes all different types of aerosols from the anthropogenic and biogenic categories, which are listed here. And then we added pollen to the model. So we used our pollen emissions that I talked about in the first half of the talk. We add them into the model, and then we add them into the different aerosol modes. This is the size distributions that I also started with. And we put the primary particle into the coarse mode. And then we put the accumulation, as we start to rupture it, we put it into that smaller accumulation mode. And we tested a couple different ways of rupturing this within the model. One where we just emit the primary pollen and we don't allow any rupture. And then where we allow just rupture at the surface, rupture in the atmosphere, and then also looking at that possibility of lightning rupture. And so we ran these simulations over the central part of the United States. So this is, it's a little hard to see here. Here's Oklahoma. Part of the reason for that was that we were funded by the Department of Energy, and they have a large field site there that has a lot of atmospheric radiation measurements, which were helpful for evaluating the model. So the primary pollen grain emissions from the emissions model I described in the beginning sort of shows on the eastern side of this domain, we have our largest amount of primary emissions. And that's really where most of our forested regions are occurring. The yellow and orange are sort of representing crop and grassland regions, which have some emissions. This was in April. And so if you think back to that time series of how the different types of pollen peak at different times, we were really trying to conduct these simulations during the deciduous tree peak, as well as the time where we saw severe convective storms. And so when we look at the simulation, so we were doing sort of an event-based analysis of a large convective event. And the idea behind selecting a large convective event would be that you'd have this sort of big, deep convective storm. And the reason that's interesting from the cloud perspective is it allows a lot of those pollen grains to be taken up into the cloud. And so we can kind of see how far they get under these very strong, deep convective conditions. So this is showing you, so this is 3 different days of the simulation every 2 days. So the first row is the primary pollen that we emit from the model. The second is the secondary pollen, or the SPP, or the rupturing pollen. And then finally is the precipitation. So in the beginning of the simulation, we don't have a lot of precipitation. We have a lot of primary pollen. We start to see a system evolve on the 2 days later. This is a large sort of strong line of convection that's coming right here. And then we start to see the secondary pollen get ruptured at the expense of the primary pollen itself. And by the time we reach the end of the simulation, when the convection is the strongest and our precipitation is the greatest, we've created a lot of subpollen particles, and a lot of our primary pollen has been reduced because we've been rupturing it in the atmosphere, as well as removing it by wet deposition. So now I'm going to change the angle here. Those were spatial plots. Now I'm going to look at vertical plots. So in this case, my x-axis is the time series. So before I was showing you think it was the 13th, the 15th, and the 18th of April. And now I'm looking at these versus altitude. So this is going up in kilometers. Just to give you an idea for these different types of rupture experiments, how far the pollen can be mixed into the atmosphere. Again, we sort of went into this thinking that most atmospheric scientists think the pollen is too large and it's really hard to get it in the atmosphere. And what these simulations show is that even despite the size, we can still have a lot of relatively high concentrations of pollen throughout the boundary layer. So in the first case where we didn't look at any rupture at all, here's our primary pollen. You can somewhat see the diurnal cycle. And then our large convective event is on this day. And in this case, we can see primary pollen being taken all the way up into the upper troposphere over 10 kilometers, which is pretty impressive. And it was a very deep convective storm. When we start including some rupture, here is only rupture at the surface, and here's where we include atmospheric rupture. You can see the primary pollen concentrations start to decrease in these plots at the expense of increasing secondary pollen, which is these shown right here. So when we have a lot of atmospheric rupture, you can see our within the whole lower part of the troposphere, the first 5 kilometers of the atmosphere, we have fairly high concentrations of the secondary pollen. And under our deep convective event outlined here in red, we can actually also see some generation and rupture that's occurring in the clouds aloft. When we do the lightning rupture, we have trouble generating a lot of the secondary particles. And so as a result, our simulations suggest that the humidity rupture may be the more important process. We can also then look, and this is I'm going to go over this a little bit quickly just in the interest of time, but if you're an atmospheric scientist, you might be interested in this, is we could also see changes, these sort of similar changes in the cloud hydrometeors, or the different types of particles. So here I'm showing you the cloud water, and this is the baseline simulation on the left for these different simulations, and then the change. And essentially, we're reducing that cloud water in the lower part, and we're creating more precipitation. And we're also changing some of these cold phase hydrometeors like graupel and snow because of these sort of balancing effects that we often see. And these changes are on the order of about 5%, which again is not, in terms of the cold clouds and in terms of the cloud water, it's not major, but it is something that we should be thinking about with respect to simulations, and it can affect the life cycle and simulation of precipitation within the models. So one of the reasons we're also interested in this specific event, and I bring this back to some of the health concerns about pollen, we might think about pollen as having just this sort of nagging chronic effect of of chronic allergies. Like, we can think of it as like a chronic symptom. And if any of you are allergy sufferers, right, the impacts of that. But there's also this other more acute effect of pollen, which is something known as thunderstorm asthma, which you may not have heard before. And so starting in the 1980s, there are several papers that popped up in the medical literature citing these cases of where after a thunderstorm, all of a sudden emergency rooms would be flooded with people having asthmatic attacks. And they started coining this term thunderstorm asthma. And it's been attributed, interestingly, to pollen. And so the idea is that what thunderstorm asthma is that we might have our whole grain pollen, it gets swept up into clouds when we have storms coming in. Kind of the rupture processes I've been talking about, it'll break it into smaller particles. And then you might have cold, dry outflows that are taking the pollen down to the ground level. And again, now people can breathe these tinier particles into their lungs, and it triggers a more asthmatic response than you might just get from your regular allergy season symptoms of like a runny nose or watery eyes, right? Because you have now all of a sudden not these large particles that are going to get trapped by your upper respiratory defenses, but you have these tinier particles that can make it all the way down into your lungs. And this is a schematic I've taken from Australia because in 2016, there was a huge thunderstorm asthma event in Brisbane, Australia, that led to several thousand people coming in and flooding the emergency rooms. And about 8 people actually died as a result of this really strong event. And so there's been a lot of interest recently in terms of, okay, well, how can we predict these types of events? And also, do we really understand what's starting to happen with them? So with the simulations I just talked about, we've been trying to understand where these processes could occur. And so this is a cross-section of one of those storms. So this is now giving you like a longitude cross-section across that Southern Great Plains region with altitude. And the pink contours are that primary pollen. So you can see generally, most of them tends to be sort of confined to the first couple kilometers. The color contours are the subpollen, or the tiny pollen particles, and we can see a lot of those in the deep convective storm. We can see this updraft that is taking particles up, as well as rupture that's occurring in the thunderstorm anvil. And you can see this sort of outflow that's occurring where that's taking the subpollen particles with it. In our simulations, which are relatively coarse for clouds, we can look at sort of the downdrafts, again, this idea that cold downdrafts are pulling these particles down. And we can see that sort of on the front side of the downdraft, we don't see a lot of movement of those particles down to the surface. Where we probably see a little bit more of it is on the rear side. And so we're currently in the process of doing some higher resolution simulations to see if we can understand the dynamics of this storm a little bit better. The last thing that I'll talk about is sort of these ice processes, which is something we've been really interested in lately in terms of trying to understand how pollen could act as a freezing, either through any of these different freezing mechanisms I'm showing here. And so this took us back down to Texas. We went back down in 2021, again, to work with Sarah Brooks. Here is us. We're in a ragweed patch at Texas A&M. This is giant ragweed. So giant ragweed grows to be like 6 or 7 or 8 feet tall. And so we collected ragweed. And this is with Sarah's on the right. And this is graduate student Brianna Hendrickson, who designed this really, really cool chamber, which we're really excited about in this study. And so this was based on some of these original medical studies where people were trying to understand how many tiny particles come off of ragweed. And so Brianna built an environmental chamber. So here's our ragweed that we picked out here, placed in the chamber. And then that allows us to look at conditions in the chamber and try to develop some new ways of a better way of generating particles than these sort of like, mixing them in water, as I showed before, because we think that this is a better representation of what really happens in the atmosphere. So Brianna's paper is currently under review, so I'll just kind of go over some of these quickly. But what they can do is we tested 3 different types of ragweed: live oak, ryegrass, and ragweed. And so within this chamber, we can turn the fans on or off. And one of the things that you can see is whenever we turn the fans on, we get this large burst of these different particles. And it was even greater for oak and ryegrass than it was for ragweed. So that allows us to sort of understand and figure out how many particles we're getting off of these particles and off this chamber. And this gets at one of our uncertainties, which is how many particles we're making, how many tiny particles are we making per grain? And so from Brianna's study, we use particle counters to calculate how many times or how many particles were coming off of each of these little bursts for the different types of pollen. And then we scaled that up to figure out the number of SPPs we had per pollen grain. And so the interesting thing is these new laboratory studies tell us before we were between 1,000 and 1,000,000, and actually the numbers came in at around 10 to the 5 SPP per grain. So the laboratory studies suggest that these higher numbers are probably a little bit more appropriate for real world conditions. The second thing that they've done in the lab is to try to look at what temperatures pollen will nucleate ice. And so again, like this gets into a little bit of details of atmospheric science, which might be more than some of you are interested in. The answer is it is a good ice nuclei, but interestingly, the primary pollen, which is shown as the circles and the triangles here, are better ice nucleators than the secondary pollen, which are shown as these squares. And so essentially, as you get to warmer temperatures, if it freezes at warmer temperatures, that means it's going to happen lower in the atmosphere, and you'll see more of that freezing occur. Whereas if you need to get all the way to like 32 degrees, negative 32 degrees Celsius, that means you have to get pretty high aloft. So being able to freeze at warmer temperatures is generally being seen as being a better ice nuclei. So then we included all of this in the microphysics within the model. So we included all these processes and we included the pollen. And again, I'm going to just kind of go over this quickly because I think this might be a little bit more atmospheric science than people are wanting. I want to spend a little time to wrap up. We did that same model domain at higher resolution, and we can start to look at this is another example of like a curtain plot. So it's time on the x-axis and height on the y-axis, and it allows us to compare our control simulation with when we only include primary pollen, when we include these ruptures, and when we include some of our new laboratory data. And so the answer is we can generally see increases of especially these higher altitude ice changes of about 50%. And we can see also the suppression in the clouds at the lower latitude. So we can see these feedbacks with hydrometeors. So in terms of like advances we've done to date, we've developed these pollen emissions modeling diagnostic tools, and we've really been working at trying to incorporate some laboratory studies into models because it'll give us better process level information. And finally, we've shown that with these couple models, that pollen particles can influence clouds, especially when we're including new ice properties. And finally, I'd just like to close with a couple emerging directions. As a result of the modeling work, we've been working with Jordan Schnell, who's at NOAA, and he works on the NOAA Global Air Quality Forecast. And so we've been trying to include our pollen emissions within the NOAA RAP Chem Forecast, which provides the air quality forecast for the United States. So right now we have it in their forecasting model, and we're currently evaluating how that model's doing. And we're hoping that this kind of provides a better tool for pollen forecasts that we think is a little bit more physically based than some of the very sparse data I talked about in the beginning. Secondly, we're seeing improved observations. Again, I mentioned that sparse data. That's a huge constraint as we try to understand this problem. There's a growth in automated pollen sensing. And so we're starting to see new techniques for people using machine learning algorithms to identify different types of pollen, as I'm showing here. And we've also been working on using LiDAR as another method. We can detect fluxes of a forest. This is data that we took at UMBS several years ago, where we can actually think we can see pollen plumes coming out of the forest. And then finally, like in terms of how we can actually measure aerosols, there's some new techniques that people are starting to use as well that go outside of like a traditional pollen count station. One is known as the WIBS, which uses fluorescence to detect biological particles. And then there's also really fancy techniques that we've been working on using like what's known as metabolomic analysis, where you're using very high resolution GC mass spec to try to develop different fingerprints for different types of pollen. And we've had some success with that. So finally, I'd just like to put up some acknowledgements for funding. I definitely want to thank this is work presented by two graduate students, one Matthew Wozniak, who graduated 2 years ago, and a current graduate student, Ingjia Zhang, as well as a former postdoc, Talman Asuba, who's now at Brookhaven National Lab. And sorry if I went too long, and I'm happy to take any questions. Thank you.
Thank you so much, Dr. Steiner. Not too long at all. I will open it up. We have plenty of time for questions from the group, and then we'll move towards graduate student questions at 3:30. So yeah, feel free to unmute if you're in a spot to do that and ask any questions, or I'll monitor the chat as well if people want to type them there.
Well, maybe I'll ask a question, Allison. I don't understand anything about pollen, so forgive me if this is completely stupid. And but I think you were saying in your initial simulations, the climate simulations, that you were trying to tune basically the beginning of the season and the end of the season using the Gaussian curve, and you were tuning those parameters based on temperature, I believe, of the year before or something like that. And I was just curious if maybe there's other factors, because again, based on experience, it's not just temperature that tells me how much pollen there is. There's might be rain the week before, or how cold the winter was, but not necessarily the yearly average. So I was wondering if you can comment on those potential dependencies.
Yeah, no, you're absolutely right. And initially when we started, we were trying to think if we wanted to use multi-factor kind of regression analysis. And the interesting thing is the precipitation data is really complicated. Like as soon as you start bringing that in, because there's kind of two effects of precipitation. One is like the immediate, like the short-term effects, which would be like, if it rains, you're going to remove your pollen. But then there's the long-term effect in terms of like drought. So if the plant was in drought in the prior year, what would that do to pollen production in the next year? And the answer is nobody knows. It's kind of amazing, actually, that we don't really know anything about this at all, because the data is so sparse and there's so little out there. There have been so few long-term continuous studies. So what we found actually when we did the regression was actually the prior year annual temperature actually did a pretty good job. And so that was why we went with that regression. Now, why the prior year? The idea, like what we were thinking was essentially like, so a plant finishes its growing season, it goes into senescence. And so, when it's growing, the interesting thing is the pollen comes out before the leaves come out. You'll start to see this now, actually, like if you go out and look, you'll see the pollen structures actually come out. For most trees, they're coming out before the leaves. So there's some sort of, not to like, I don't want to make it sound, no, I don't want to anthropomorphize it too much, but like, there's a decision, an allocation decision that's made to say like, okay, this is how much pollen I'm going to make. These are how many resources I'm going to put towards pollen. And so that seems to be, so we're sort of thinking like, well, prior year must inform potentially this sort of upcoming year, which is why we made the decision to go with that. And honestly, like the regressions kind of look best using the prior year annual average temperature. We tried different things like fall temperature, spring temperature, winter, we tried like all these different things. And we also also tried using different precipitation metrics. I still think soil moisture is also an important factor, but we have so little data that it's really hard to say at this point. But yeah, no, it's a great question. It's something we hope that eventually like we'll get better data on to be able to do better.
And was temperature also the driving factor in the simulations of the future climate too? Was that basically, because you were showing those animations of when the pollen would come out,
yeah.
Do you think that was mainly temperature driven then?
Yeah, so it is like we have the short-term precipitation effects in there. So essentially, like if it rains, we allow that to change the daily pollen emissions potential. And then when we couple it in the model, we also have a wind. I didn't say this out loud, but I probably should have. We have like a wind-dependent factor and like precipitation and a relative humidity factor that control like the timing of the release. But like in terms of long-term precipitation, that's not included because when we look at the data, it's just so noisy, you can't really tease out any relationship. So in terms of the climatic factor, there is precipitation, short-term precipitation effects included, but the dominant driver really is temperature.
Time for a few more questions.
I can keep going, so I'm going to keep going. So in the laboratory experiments, you collected those coarse pollens and then you put them in the vials and then I guess you shook them.
Yep. Mm-hmm.
And the intention was to break them up, right? So to
yeah, and you don't even really have to, we did multiple tests, like you don't even really have to shake them. You can just put them in water and like stir it up. And we did like time tests. We did like, 10 minutes, 20 minutes, 30 minutes, an hour. It didn't matter. It actually happens really, really quickly. So it didn't seem to be a function of time, which is why we think it's kind of like a cloud relevant process.
That's so cool.
Mm-hmm.
And then you were saying that when you did the fancier method with the plant in the chamber, you got results that were even higher, an even higher count, right?
Yeah, I mean, so it's, yeah, the one thing that's challenging, like experimentally about doing, like, we often call them like the wash water experiments, being like we put the pollen and we'd wash it. And then you take that water and you atomize it. So when you atomize the solution, it makes tons and tons of particles. So you don't really know like what actually would be like equivalent to coming off of a plant. So the chamber experiments allowed us to do was say like, okay, if you stick a plant in there, like if you have a branch with this many pollen catkins hanging down, how many SPP can it make? And that was what the chamber study really allowed us to do in a way that the wash water experiments don't really let you get to.
I'll be quiet.
Thanks for the questions.
I was wondering, though you made mention of the scale, it's actually at a much coarser scale that you use. So I was wondering, is the scale a major requirement for the WRF-Chem model simulation? Does it really like influence the output of the simulation?
I'm sorry, you said the what part of the simulation?
The scale that you use for the emission, The scale? Yeah.
You mean like the model resolution?
Yeah, the scale, the model resolution.
Yeah, so I mean, from the pollen emissions perspective, well, generally with emissions, better resolution is better, because you can get finer, more, you can resolve different features better. Generally, we're limited by the land cover resolution that we have for the forest data, which is 12 kilometers. So that's as good as the land cover data we had to do the pollen emissions model. So we ran the WRF model at 12 kilometers also, but for clouds, 12 kilometers isn't really that great. So as you want to, if you want to be able to resolve clouds a little bit better, we usually like using something like a 3 kilometer or a 4 kilometer resolution. And those are the ones that we're doing the ice nucleating studies with now. And the reason for that is just because you have to, it's, it sort of depends on your perspective of how well you want to resolve clouds. But generally, it's sort of thought once you get to about 3 or 4 kilometers, you don't need a separate convective parameterization to represent clouds. But if you are at 12 kilometers, then you have to have both the microphysics and the convection. And so as we look at these sort of aerosol cloud interactions, it's really better not to have a convective parameterization. It's really better to kind of focus on the finer resolution processes where you can just run the cloud microphysics. So there's kind of 2 parts to your resolution question. Hopefully I answered that.
Yeah, that's fine. So the inputs to the model, is it you rely solely on the forest data? How did you take account of other other areas or the regions of the countries where there may be some small amount of trees and so
yeah, so for the tree data, we've been relying on US Forest Service data. So the one thing that's good at is like large forests. What it's not good at is urban areas and suburban areas. The other thing is we focused on the United States because we did have tree data over the US, at the sort of level, like at the genus level, which is what we were looking at to be able to resolve the pollen better. That data is harder to come by globally. And so that's why we also developed this plant functional type method. So in theory, you could run it within a global model as well.
Oh, okay. That's nice. Thank you.
Mm-hmm.
Thank you so much, Dr. Steiner. Again, I'm going to invite the faculty to exit and leave the graduate students to ask any questions about pollen, about other research, about professional development, but we do want to thank you so much for your time this afternoon, and I'll leave it to the graduate students to finish things up.
Thanks for coming. I'm gonna stop sharing. Is that okay?
Okay.
Yeah. Okay.
Yeah.
That way I can see you all better.
So thank you very much for the presentation. What will you recommend as the best approach to model aerosol? Not necessarily like pollen aerosol, like if you have interest in modeling aerosol, for instance, is there any?
Yeah, I think it depends. So I think the model selection is really a function of what kind of question you're going to answer. So for some people, like the aerosol question might be related to climate. So maybe you're interested in, what is the aerosol forcing and how is that going to influence climate? Or you might have a question that's more about air quality, for a more specific region. So like kind of 2 questions is like, what is the intent? Like, I think one is because that'll give you an idea of how much chemistry you need to have in the model. So like what I mean by that is generally the chemistry models are computationally expensive to run. And so there's a lot of variability in the complexity of treatment you can use for aerosols. WRF-Chem tends to be on the more complex side. We were trying to really get at this balance of biogenic versus anthropogenic aerosol. And so that's one of the reasons why we used a more complex model. If you're trying to do urban air quality, then you want to be doing higher resolution and you also want some chemical complexity. So I'd say it kind of depends on the question that you're after. Another thing I'll say is like, we're running WRF-Chem, which is coupled chemistry and meteorology. That allows us to look at if you change the aerosol, the clouds will change. Another way of doing atmospheric chemistry simulations is using chemistry transport models, where you're really just focused on the chemistry and then you're giving meteorological boundary conditions like from reanalysis or something, and you're uncoupled. So it might mean that like you might simulate aerosols, but they wouldn't interact with clouds. So I think it kind of sorry, that's a long answer to your question, but I think it sort of depends upon like, what scientific question you're trying to ask and then figuring out the right tool for that question.
Yeah. Thank you. So you talk about WRF-Chem being very expensive. So like, what model will you recommend if you are like modeling air quality, urban air quality?
Yeah,
So-
I mean, I think, you know, one of the, so there are like, so you can run, well, it depends. Like, so, you know, sometimes you can run like an uncoupled model like CMAQ. So the EPA model, the EPA air quality model is called CMAQ. It stands for Community Mesoscale Air Quality Model, AQ. That's what the AQ is. And so that is a little bit less intense to run because it doesn't, you use meteorology from a different model. So you might run WRF and you generate the meteorology and then you feed the meteorology to CMAQ and then you run the chemistry. So it's almost like it's a two-step process. And that makes it a little bit less computationally intensive to run, but then you can still do high resolution simulations to do like urban air quality.
Okay. So like if you are modeling urban air quality, is there any way you parameterize the other type of aerosol? Like you are trying to consider aerosol, let's say from biogenic emission, not from biogenic, from anthropogenic emission. So how can you like you know, ensure that you are specifically modeling aerosol from anthropogenic emission?
Yeah, so usually there are options within the model, like you can turn
Okay.
different things on and off. So you could turn off biogenics. You know, the anthropogenic, again, like some of the chemistry gets rather detailed. So some models do it more simply than others. And so when you run something like CMAQ or WRF Chem, they have different options for like what kind of aerosol model do you want to use? What kind of gas phase model do you want to use? It's like those changes, you can pick ones that are a little bit more computationally efficient.
Okay. Thank you very much.
Mhm.
Thank you. Thank you.
Yes, no problem.
Hello, ma'am.
Hi.
I thank you for your presentation. It was very interesting. I kind of have a general question about regional climate modeling. I'm currently learning it for my research. So I was wondering what was your process of using regional climate modeling in your presentation today? Is there a paper that you can link us to so we can read it and see it? Because I would love to look at your methods.
Yeah, so the first one I mentioned, like the first modelings today I mentioned where I talked about those precipitation effects, that was using RegCM, which is a regional climate model. So that was, the first author was Wozniak, Woziak. And it was a paper in Geophysical Research Letters in 2018. And so that talks about the methods we used with RegCM to do the pollen. You can do like, you know, so, you know, but there's lots of other RegCM studies that I've worked with before. So some of those are like on my website about how we set up the model, you know, in terms of like conducting length of simulations and things like that. The one that I mentioned today was pollen. We did what we called like multi-member ensembles. So instead of running for like 20 years all in a row, because we were looking at the spring and we were trying to do multiple versions of the same run, which is useful sometimes when you're dealing with aerosol cloud work because it can be very noisy. It's often good to have what we call like a multi-member ensemble, which means that you run the same simulation several times with slightly different initial conditions, and then you average the output, and that takes out some of the noise of the simulation. So that's one technique people often use to kind of come up with more robust results.
Thank you.
Mhm.
So I have more of a general question. So what skills did you like learn in your PhD or postdoc that you carry over that are totally like general skills?
Yeah, I mean, generally your PhD time is when you're really trying to do a lot of skill building. And so, you know, again, so I would say when I was a graduate student, I spent a lot of time learning how to edit and run different models. And so like doing a lot of coding, that was something that I spent a lot of time doing, learning how to do data analysis and data visualization. Those are things I still do all the time. And then as a postdoc, I actually worked a bit with a group that did experimental work. And so while I don't really do that work, like I talked about some collaborations we have, some of the stuff I do in the lab, but like in my research at Michigan, we don't actually have a lab. I collaborate with people with labs. And so sometimes, but it was helpful in terms of like learning about how people use different techniques to answer different questions and problems.
Right. So you're kind of looking at how different methods can be transferred to different problems.
Yeah.
Just I jump in here. Thank you, Professor, for the wonderful talk. Just continue from the last question. Do you think that this learning stops anytime now?
No.
Still learning?
No, I'm always, I mean, I still really enjoy going to talks and hearing, you know, like even student presentations. I learn a lot when I'm on different doctoral PhD committees and learn about my colleagues' work. And, you know, that's one of the reasons why you have like other professors come in, is because they bring different areas of expertise and can kind of bring different perspectives. So no, it's always ongoing.
Thank you.
Sorry, I have another question.
Okay.
Yeah. So will you recommend using remote sensing for stuff like maybe modeling aerosol emission? Because I understand that you can use remote sensing to like model this aerosol optical depth. So do you think it's like a viable option, more especially if you are like studying regions where there are no data?
Yeah, so for sure, remote sensing data can be very useful in terms of like getting spatial information where we don't have like again, like these sparse networks, or if you're looking in different parts of the world where we don't have any ground-based observations, they're great. One of the challenges with aerosols and with remote sensing though is remember that you're getting, you know, what you actually are retrieving from the product, usually, you know, you have a satellite and it's usually doing some kind of technique, you know, some optical technique that's retrieving an entire column, right? So for example, like if you're thinking of aerosols, like one of the, you know, the most common satellite products is aerosol optical depth.
Yeah.
Right? And that's giving you generally a total column concentration. So sometimes it can be difficult to back that out directly to like an aerosol emission per se, or it's hard to attribute. You know there's aerosol there, but you don't know what kind and you don't know what size. So like what people have been doing is they've been using some of that data in conjunction with models to, you know, come up with better PM estimates and emissions in regions where we don't have data. But it's always going to be this like blend of models and satellite data, you know. So, you know, the satellite data can be useful. It can be also used to evaluate models, right? You could run a model simulation and say, okay, my model says there's aerosol here. How does that compare with, you know, aerosol optical? How does my model aerosol optical depth, which is taking into account all those complicated processes, how does that compare with the satellite optical depth? That's another way people use it as an evaluative tool. There are other satellites, you know, like if you look at Calypso, that's one of the ones from NASA that does vertical profiles. So in that regard, you can get more information about the vertical structure of aerosols. They do some aerosol classification as part of that. You know, again, like still it's more general, like they'll say like it's anthropogenic or it's biomass burning or it's dust. Based on the optical properties that they're using from that specific satellite. So there are some ways to do it, but you always want to be a little bit careful. But I think, you know, ultimately it's a great resource in terms of getting data where we typically don't have data.
Yeah. So in terms of like maybe modeling using the remote satellite data, so at what height, since I understand the satellite actually gives out the vertical distribution of the aerosol, so at what is the minimum height if you are trying to like infer the surface aerosol concentration?
Yeah, so that's challenging, right? Because even Calypso, which gives you a vertical profile, doesn't really give you a concentration per se, right? It's giving you some information about the optical properties in the column. And again, that's where you really need the model to connect that back to like a surface observation. And so like that's where it's really, I would say it's like a fusion of the model data and the satellite data to be able to back out a surface concentration.
Okay, thank you very much. So one last question. Apart from aerosol, what other atmospheric pollutant will you do you think is easy to model using remote sensing apart from bioaerosol?
Well, the new one is NO2, nitrogen dioxide. So actually just last night, they just launched a new satellite. NASA just launched a new one called TEMPO, TEMPO. And NO2 is like in the US, it's a criteria pollutant. It's also a precursor for ozone. Ozone's harder from remote sensing because you have so much ozone in the stratosphere that it's hard to separate the stratosphere and the troposphere. But NO2 is one that we've gotten very good information on from satellites. And you can really see, again, that's a column concentration. So then you have to use a model to back out a surface concentration. Formaldehyde is another one that's been rather successful in terms of, that's not a criteria pollutant. But I would say, like, from the remote sensing perspective, people have had a lot of success with NO2, and that's an important precursor for other, for ozone and also for PM.
Okay. Oh. Thank you very much.
Sure.
Yeah.
Okay. Hi. I have more like a general question. As I was watching your presentation, I was thinking, like, is there any air quality standard for bioaerosols, like federally? And if there is any threshold for bioaerosols, this. Do they give any alert or seasonal outlook for bioaerosols, like in this season it will be hazardous or something like that?
So not really. So so it's not federally regulated at all. So what the US regulates is PM 2.5 and PM 10. So it might be some of the aerosol would, some of the pollen might come under PM 10, but in general, we don't. It's really not regulated. In terms of forecasting, no. Like in terms of seasonal forecasts, again, no one's really developed a good seasonal forecast tool, and that's something that I think we're hoping to change. And it's honestly, it's kind of surprising because we haven't had that done yet.
Yeah, thank you.
No other burning life questions?
Yeah. And we're also right out of time. So
Okay.
Any last questions? I guess there is no any other question.
Yeah.
Well, thank you very much for having me. If you have any questions about things I've talked about, feel free to follow up with me by email.
Okay, definitely.
Thank you.
I'll
Thank
do
you
that.
for
Thank you.
so much.
Nice talk.
Thank you for the presentation.
Bye. Have a good weekend.
Thank
You too.
Bye.